All right, so welcome to the sixth and last lecture of the physical biology module. Let's start by quickly having a look at what we did in the last lecture. So in the last lecture, we were talking about proteins and their structures. And what I told you that if you look at 1,000 different types of proteins that we have, all of these proteins are made from only 20 different types of amino acids. Okay, so we have 26 alphabets in English, and if I give you the sequence of letters written on a page, by looking at the sequence you can tell me what it is: song, poetry, speech. Okay, but if I give you a sequence of few amino acids, by looking at the sequence of amino acids you cannot tell me what will be the three-dimensional structure of the protein. So given a sequence of amino acids, the can we predict the three-dimensional structure of the protein with accuracy is still a unsolved problem, because we don't know exactly the rules according to which protein acquire the three-dimensional shape, and we do some physics based calculations to predict the structure of the proteins by doing computer simulations. Okay, and those simulations are called molecular dynamic simulations. I will talk about those after a few slides. Then one of the things which I told you, that whenever we are talking about protein folding, the proteins are free energy minimizers. Okay, because the folding is happening in presence of thermal energy. There we looked at the concept of microstates and macrostates. So what I told you, that macrostates are like global states and microstates are like local states which belong to a given macrostate. So a given macrostate can consist of many possible microscopic states. And we looked at the relation between between enthalpy, entropy, and free energy. And then we also looked at the relationship between the free energy and the partition function. And we also learned how to calculate the entropy from the knowledge of the number of microscopic state that can belong to a given macroscopic state. And finally, we looked at a very simple model of protein folding, which was HP model of protein folding, where we classified all known amino acids into two groups. One of them was hydrophobic and the other one was polar. And then we use this toy model of protein folding to look at one of the numerical problems, in which state I will find a given protein with the given sequence. Okay. So what we'll do in this lecture, we'll look at another toy model of protein folding. Okay, so please look at this problem. What is given here, that consider a protein consisting of three amino acids A, B, and C connected with bonds of equal length. So these are three amino acids. So this is a polypeptide or a small protein. Okay, and these three amino acids are distinguishable. Okay. So this protein is placed on a square grid such that only A and C are free to rotate in a plane due to thermal energy, such that bonds are always aligned along the grid lines. Okay. So what is given to you, that this protein is consisting of three amino acids, and there are these two bonds, and since it is kept in a thermal environment, due to thermal fluctuations these bonds can rotate, and whenever they are rotating they can only align along these grid lines. Okay, so for example, A can go here, C can go here, and so on. What is also important here, that different lines are denoting different colors. Okay, I think I made a mistake, so let's assume for the time being that these three amino acids are identical. Okay, and only the grid lines are different. Yes, Priyans, you have a question? So these three amino acids are identical, ABC. So ABC is only the labeling, and they are placed on a surface where, because of thermal fluctuations, these two bonds can rotate. And if you look at the grid lines are having different colors in different directions. Okay, what it means, that the space is inhomogeneous. That means if you are going in this direction, let's say if it's an example of a room, if you are going in this direction there is a door, in this direction there is a window, in this direction let's say there is a curtain, in this direction there is a, let's say, a screen. Okay, so each direction is a unique one. And suppose if you have a protein, if you have to have that kind of environment for the protein, you can assume that protein is charged one and it is kept on a surface which is not uniformly charged. then for different orientations of the protein will have different amount of energies. Okay. So what is given here in this problem, that binding energy of the protein is given by epsilon A1 plus cos theta AC. So bending energy of this protein is given by epsilon is equal to A1 plus cos theta AC. So the energy of this protein depends on the bond angle between these two end amino acids, and which is A and C. Okay, so it depends on the angle theta AC. What is being asked here, that what is the energy of the state conformation? Okay, if this is the form of bending energy we have for this protein, and then what is the energy of the bent conformation? Okay, so now anyone would like to tell me what will be the energy of the state conformation? Anyone? If this is the problem, and if this is the form of the bending energy we have, what will be the energy of the state conformation? Anyone? Somebody give an answer. What was the answer? 0. Okay, very good. And what it will be for the bent configuration? So for straight conformation, the energy is 0, and for the bent conformation, the energy is A. Okay, and the question which is being asked here, what will be the structural state of this protein when A is small and when A is large? Okay, now if you apply the classical mechanics to this problem, what is the answer you are going to get? Which state will be preferred, or in which state you will find this protein?
Straight when A is large.
It will be found in the straight structural state, because that is having the lowest average energy or energy. Okay. However, since we know that this entire system is under the influence of thermal energy, we cannot apply simple energy minimization to this problem, because this is a problem in the microscopic world and we cannot ignore the thermal fluctuation. So instead of looking at the average energy of the system, we have to first find out what is the free energy of the system that will be in the state as well as in the bent conformation. Okay, and if we have the knowledge of the free energies in the state and bent conformation, then we can say in which state you will find this protein, right? So whichever will have lower free energy, that state will be preferred, right? However, if you apply simple classical mechanics, we get the answer whether A is small or large, it will be always the state structural state. All right, so let's see how we can work this problem out. Let me switch to my iPad. Just give me a second. Now let me share the screen. So the problem which is given to us is that this protein is there, A, B, and C. Okay, have 3 amino acids, and it is placed on a square grid. Okay, and grid lines are different in different directions. That means I can plot this one like this one something like this, the other one something like this. Okay, maybe this one like this, and this one like this. I don't know. So each direction is a different one. So what we saw, that the form of the energy which is given is A 1 plus cos theta AC. And what we found, that if the protein is in the steady state, the energy will be equal to 0. And if it is in the bent state, which means something like this, B is here, E is here, and C is here, then it will be equal to A, right? And if we have to answer this question, then what we have to find out, what will be the free energy of the straight state and what will be the free energy of the bent state. Okay, we know that the free energy of the state, macroscopic state, will be given by what is the average energy of the straight macroscopic state is minus T S, entropy of the straight macroscopic state. Similarly, this one will be UB minus T S B, right? Now, the question which I have for you, that I am calling that straight state as a straight macroscopic state. That means there are also other possibilities for this protein, right, to exist in other microscopic states. So how many different types of configurations are possible if this is the for this given problem? How many configurations we have for the straight state? Let me ask, what is the value of W for the straight macroscopic state? Anyone? In how many ways I can put this protein on this square grid in state configuration? By rotating the bond angles only by 90 degrees. So this is one configuration. What is the other possible one?
Perpendicular to it.
Please say that again. Somebody answered it. What was the answer?
He is
4.
up, B and she is down. He is
Okay.
in the center.
So there are total 4 configurations, right? For example, this is one of the possibility. Let's say A, B, and C. Okay. In the second one, what can happen? That B stays in its place, C rotates and comes here. Then A goes here. The other possibility is that B still stays in its place, A goes up, and C goes down. Then the other possibility is just a reverse of this, C stays up and A goes down, right? So we know that W for the straight state is equal to 4. However, for the bent configuration, how many bent configurations are possible? Anyone? Please raise your hand and I will ask. Those who know how many bent configurations are possible, please raise your hand and I will ask. How many bent microscopic states are possible? Yes, Dhruv, please go ahead.
8 configurations.
There will be total 8 possible configurations. I will show you shortly how we are going to get 8 configurations. Okay, so that's very good. Okay, this is considering that the space is inhomogeneous, right? Now suppose I assume that the space is homogeneous. That means all the directions are identical. In that case, what will be the value of WS and WB? Yes, Jay.
WS equal to 1 and WB equal to 1.
Okay, very good. So in that case, if the space is inhomogeneous with the same protein, it doesn't matter how I keep it. Okay, so WS will be equal to 1 and WB will be equal to 1. Okay, so let me just quickly show you how we are going to get 4 and 8. Those who were not able to figure it out. Okay, just quickly show to you. So please look at this figure that there are total 4 possibilities for the state macroscopic state, and there are total 8 different possibilities for the bent macroscopic state. All right, so Ws is equal to 4 and Wb is equal to 8. Okay, and energy of each of these state configuration is equal to 0, and energy of the protein in each of these bent state is also equal to A, right? So if I take the average of the energy for the state macroscopic state, the average energy will be also equal to 0, because all the 4 configurations have energy equal to 0. Okay, similarly for the bent state, since all the 8 configurations have same energy, the average energy will be also equal to A. All right, and therefore we can write down the final expressions for B free energies like this. Share the screen. So what we have found, that the energy of the straight macroscopic state, average energy of the straight macroscopic state will be equal to 0, because there are 4 configurations, each of them with energy 0, minus T Kb ln 4, and the value of ln 4 is 1.4, so this becomes minus 1.4 kBt. Okay, so this is the value of Gs I'm getting. Then here, if you look at this becomes, each of the configuration has energy A, and therefore average energy will be also equal to A minus T Kb ln 8, and the value of ln 8 is about 2.1. So this becomes A minus 2.1 kBt. Okay. All right. So what I have got is Gs is minus 1.4 kBt and Gb is equal to A minus 2.1 kBt. Okay, and what was given in the problem, that in order to go from the straight, when it is in the straight macroscopic state, the energy is 0, right? And when it is in the bent state, the energy is A, right? That means it requires certain amount of energy to go from straight state to the bent state, and that amount of energy is A. All right, now suppose this bending is spontaneous, that means in order to go from here to here, no energy is required. That means the value of A is equal to 0. If this bending is happening spontaneously, okay, it doesn't require any additional energy, just thermal energy is good enough. Then what we are going to get, in that case, Gs will be 1.4 kBt and Gb will be equal to minus 2.1 kBt. What it tells you, that if no energy is required to go from straight configuration to the bent configuration, or if a system can go spontaneously from straight configuration to the bent configuration, then the system will be always found in the bent configuration, right? Why? What is the reason for that? Because this is what you will not get from the simple energy minimization, right? So what this is telling you, that if this process is spontaneous, if a given process is spontaneous, for example, if the bending is spontaneous, then in that case, system is trying to go to the bent macroscopic state. What is the reason for that? How do I interpret this result? Anyone? What is the physical reason for that? I'm sorry, your voice is getting stuck. Can you please repeat again? Still, voice is very bad. Voice is, I mean, echoing a lot. I can't hear it properly. Anyone else would like to give the interpretation of this result? What is the, how do you interpret this result, that when no energy is required, if a process can happen spontaneously, then system is going to the state where the free energy is minimum? Why system is behaving like that? Any idea? Any thoughts? Please raise your hand if you'd like to interpret this result. Yes, Jay, please go ahead.
It has more possible microstates.
Yes, so what is happening here in this problem, that state, bent state has a large number of possible microscopic configurations. Okay, and since it is having a large number of microscopic configurations, okay, the bent macroscopic state also has a larger entropy. So in this case, system would like to always go to the maximum. This is what we have studied about the entropy right from the beginning, that entropy is a measure of disorder or randomness present in the system. A system always tries to go to the maximum entropy state, and here you see that if a reaction is spontaneous, then it always tries to go to the state of maximum entropy, and in this case it will be the bent state, because bent state has a large number of possible microscopic configurations or microstates, and by going to the bent state, it can maximize its entropy and it can minimize its free energy. And that's why in this particular case, if we assume that if no additional energy is required, or the energy amount of energy, let's say, required is very small, let's say 0.1 kBT, then even in this case the system would like to stay in the bent state, because in the bent state it can acquire a large number of possible configurations and it can maximize its entropy. All right, so this is what we have again learned from this calculation, that any system tries to go to its maximum to a state where it has maximum number of possible microscopic states, because then it can have a maximum entropy. All right, now this was the case for a small a, right, when the value of a is small, when there is very little energy required or there is no energy required. Okay, in that case what we found, that in this case the system will be found in the bent state will be preferred, right? Will be preferred, right? However, if we consider that the a is large, something like 1.5 kBT, a large amount of energy is required to go from the straight state to the bent state. In this case, what we'll get, that GS will be equal to minus 1.4 kBT, and then in this case GB will be equal to minus 0.6 kBT. Okay, now here if that is the case, then system would like to stay in the straight state, right? Because that is the state which will minimize the free energy. All right, so it always depends on the competition between the average energy of the system and the entropy of the system, and this is again similar to what we saw in the last example, where we studied the another toy model of protein folding. All right, any questions or comments? All right, if not, I will move ahead. Let me again share my slides. So what we have done so far, we have only considered the toy model of protein folding. However, if you look at the problem of protein folding in reality, then a given protein monomer can have many different types of possible interaction. For example, electrostatic, bending, van der Waals. Okay, and then these protein monomers can also interact with our water, and their interaction can be different depending on the amino acid. It can be a hydrophobic interaction or it can be a hydrophilic interaction. Here I considered very simple forms of energy that are enthalpies. Okay, I took very simple forms of, let's say, bending energy here in this case or in the previous example. But if you look at the energy forms of the energies or enthalpies, they are much more complicated than the simple examples that we have considered so far. And in this case, one has to worry about the entropy of the whole system, which consists of protein monomers, water, and other types of ions, okay, which are present along with the protein. Okay, so it's a very, very complex problem. And people try to study the protein folding using the computer simulations. And I said in the beginning, these simulations are called molecular dynamic simulations. Now, when a protein is formed, it is generally in the unfolded state. Okay, and then once it starts to fold, it goes through different possible states. Okay, so if you look at the protein folding, then when this protein starts to fold, its free energy starts to decrease, and it can momentarily stay in different macro states for certain amount of time. So all of these temporary states, okay, are called local minima. But when protein is in the its native state, or when it is completely folded, then it is always found in its global minima. So this is the typical free energy landscape which exists for the protein folding. And because of the shape of this curve, this this is also called the folding funnel. All right, now what I'm going to show you is a computer simulation of protein folding, which has been done on a computer only for 6 microseconds. So somebody has studied the protein folding of a small protein only over a period of 6 microseconds, and we'll see why the simulation has been done only for 6 microseconds, because all of us have a feeling that nowadays computers are very fast, so why only 6 microseconds? Okay, so the whole idea of showing this to you, that how people people study the problem of protein folding and how much computational resources it requires. Okay, what kind of calculations are involved? Okay, so let's first look at this movie. Okay, where initially we'll see that the protein was in the unfolded state and then it would start to fold. Okay, just um, so this is the completely folded structure, and how this folded structure is being obtained is shown to you. So you see that this is the completely unfolded structure, and then protein will start to fold as the computer simulation will proceed. Okay, so now this is the folded structure of the protein. Okay, starting from a completely unfolded structure. Okay, and this entire calculation was done only for a time period of 6 microseconds. That means the protein folding was seen only over a time period of 6 microseconds. So this was a simulation of 6 microseconds, but probably it took many days to calculate to complete this calculation. All right, so now how it is done. So this is typically done by using something called molecular dynamic simulation. Okay, so molecular dynamic, what you generally do in a molecular dynamic simulation, you calculate the motion of the atoms in a molecular assembly using the Newtonian dynamics to determine net force and acceleration experienced by each atom. Okay, so if somebody asks you a question, what is a molecular dynamic simulation, then you can say that in the molecular dynamic simulation we try to look at the motion of each of the atoms of a biomolecular assembly. It can be a biomolecule, it can be a protein. And what we try to calculate is the force experienced by each atom, and from the knowledge of the force experienced by each atom, we calculate its successive position. So basically, we solve the Newton's equation of motion. Okay, F is equal to ma. Now, when you have to calculate force on each of the atoms, okay, now we know that the force is nothing but it is the derivative of the potential energy, and this potential energy can come from various sources. For example, let's say if this is an example of a small molecule, I'm not showing you thousands of atoms, okay, I'm just taking an example of a simple molecule, okay, but we know that even a small protein can have at least 10,000 atoms. Okay, now suppose we have a small biomolecule, then there are different types of interactions which are possible. For example, there can be a bond stretching, right? So there can be a bonded so bond stretching is one of the bonded interaction. Then the angle between the bonds can change, and that requires some energy. Okay, so that is called angular energy. Then one bond can rotate with respect to the other bond, that is called torsional energy. So we calculate the contribution from all types of bonded energies, and then there are also non-bonded contributions to the energy. For example, there can be a van der Waals interaction between two unbonded atoms which are far apart from each other. If the atoms are charged which are not bonded together, they can also have an electrostatic interaction. So what is done in a molecular dynamic simulation? Let's say if you have thousands of atoms, you calculate how much force each of the atoms are feeling. Okay, and generally the starting structure, your starting structure is the structure which you obtain either from the generally you obtain it from the experimental techniques like cryo-EM, X-ray crystallography, or NMR. Okay, and sometimes people also model these structures based on sequence similarity. Okay, so once you have the atomic coordinates from, let's say, from a crystal structure, the molecular dynamics simulation programs calculate the force experienced by each of the atoms, and this is how it is typically calculated, by taking the contribution of bonded as well as non-bonded interactions. And once you have determined the force experienced by each of the atoms, what you do, you start with some initial position which is typically determined from the initial structure of the protein that you have. Then you choose a small time delta t. Okay, now the whole idea is that once you know the acceleration, you can integrate it over a certain time period to get the velocity, and then you can further integrate the velocity to calculate the next position. And there are various methods to calculate the successive position from the knowledge of the acceleration. Okay, so most of the molecular dynamics program do the same thing. They first calculate the force experienced by each of the atoms, and then they calculate their successive position, and then you keep doing calculation as long as you want to see the movement of the atoms which are there in a biomolecule. Okay, so as I said that the time period which has to be chosen for doing these kind of computer simulations is extremely small. These calculations have to be done at a very small intervals, and these intervals are typically of the order of femtosecond. Okay, now I will not go into the details of why you require such a small time step. Okay, but please take it granted as of now that we require a very small time step to do these calculations, because this is one of the requirement for the doing the correct computer simulation. Okay, um, so once you have the knowledge of the forces, from the knowledge of the forces you determine the acceleration, and from the knowledge of acceleration you calculate its successive position by using certain algorithms, and then you move the atom to its next position, you increment the time, and you repeat this calculation for the period you want to do this computer simulation. All right, now I have a problem for you to work out. Okay, so suppose, let me share my slides. So what I'm discussing right now is the MD simulation, molecular dynamics simulations, right? And what we are doing in a molecular dynamics simulation, suppose there is a biomolecule, Okay, we are calculating how much force each of the atoms are feeling, and from that knowledge we are trying to calculate its successive position. Okay, by solving a number of equations. So suppose I am trying to do this calculation for a biomolecule which has 1000 atoms. Okay, but we know that generally even a small protein can have 10,000. So let's say this protein for which I'm trying to do calculation is having 1000 atoms, and I know that when I have to do this calculation has to be done at an interval of 1 femtosecond. Okay, so let's say 10 to the power minus 15 seconds. Okay, now from the knowledge of the force experienced by, let's say, each of the atoms, this is first atom, this is second atom. Okay, from the knowledge of the force to calculate what will be its next position, let's say, x1 after time delta t, I have to do, let's say, 10 calculations. Although I'm taking a very conservative estimate, there can be at least 100 calculations. So let's say position calculation of successive position of each atom requires at least 10 calculations after you have calculated the force. Right? So question is, I have a protein which is having 1000 atoms, and calculation of, to calculate the next position of the atoms, I have to do at least 10 calculations at each time step, because simulation starts from t is equal to 0, then I have to calculate the position at time delta t, then I have to calculate the position at time 2 delta t, 3 delta t, and so on, right, at regular intervals. And in order to go from here to here, I have to do 10 calculations. From here to here, I have to again do 10 calculations, and I have to do these 10 calculations for each of the atoms in the molecular assembly. Now suppose I want to look at how the different atoms of this biomolecule are moving over a period of 1 microsecond. Okay, if I want to do a computer simulation of this biomolecule using MD simulation for a total time period of 1 microsecond, where I have to choose the time step of delta t of 10 to the power minus 15 second, there are 1000 atoms in this molecular assembly, and calculation of successive positions requires at least 10 calculations. Total, how many calculations I have to do? Please calculate, and I will ask. So for 1 microsecond of simulation, how many calculations my computer has to do? Please calculate that and raise your hand. If the question is not clear, I can again repeat. So what I want to know that, for this small biomolecule which is having 1000 atoms, if I have to do a computer simulation for 1 microsecond, given the fact that calculation of successive positions required at least 10 calculations, how many calculations I have to do? Okay, Fajan.
10 to the power 13.
10 to the power 13. Okay, anyone else? Yes, Jay. Do you agree with that answer?
Yes.
Okay, very good. So we have to do at least 10 to the power 13 calculation. Adit, I think you also agree with that, right?
Yes, sir.
Right. So, if you look at the number of calculations, right, the total time period for which I have to do is, let's say, t, then this becomes t upon delta t. Okay. So this is something like 10 to the power minus 6 divided by 10 to the power minus 15. This gives me a 10 to the power 9 times. I have to repeat this calculation for each of the atoms. Okay, so for 1000 atoms, this will be 10 to the power 3. This entire thing will be multiplied by 10 to the power 3, and for each of the atoms I have to do 10 calculations. So that makes it 10 to the power 13 calculations. Okay, So you can see that even for a very, very small biomolecule, I told you typically there are 10,000 atoms. But let's say even we take a very conservative statement, estimate that there are only 1,000 atoms. For doing computer simulation of something over a time period of 1 microsecond, for even for this kind of small protein, I have to do at least 10 to the power 13 calculations, where I try to oversimplify things. I said that it just required 10 calculations, but actually the number of calculations are more than that. Okay, so we have to do 10 to the power 13 calculations. Okay, and this is why molecular dynamics simulations are very, very time consuming. Okay, sometimes we have to run our simulations for months to get the data. Okay, so even with the computers that we have, supercomputers that we have today, one can go up to the time step of, let's say, milliseconds. Okay, but still looking at the dynamics of a biomolecule over a period of seconds or few minutes is completely out of question if you are trying to do the atomistic simulation of biomolecules. Okay, so although we have a feeling that computers are very fast, they can do calculations much faster, but for doing these kind of scientific calculations or molecular dynamics simulations, whatever computers we have, they are still very, very slow. All right, for example, in our lab we can typically go to 100 nanoseconds to 100 nanoseconds. There is also super computing facility at IIT Bombay. We can go to even slightly higher time for slightly higher time, but even on these supercomputers we cannot run our calculations for months, because there are other users who have to use these computers. So there are a lot of limitations, and the whole point of telling you all of this is that people do study the folding of the proteins or the structural changes in the biomolecules by doing the molecular dynamic simulations, and these molecular dynamic simulations are very, very time consuming because of the number of calculations involved. All right, okay, so let me share my slides again. So when I'm talking about the protein folding, and I do not take the name of Professor G.N. Ramachandran and his contribution to the field of protein science, it will be a complete injustice, okay, to Indian science. So what Professor Ramachandran and his colleagues did, who were working at ISC, Professor Ramachandran and his colleagues said that due to various constraints of arrangement of atoms in 3D, neighboring amino acids or protein monomers in a protein cannot fold into any shape, and there are some constraints their arrangements have to satisfy. Okay, so let me turn on my video for a second. For some reason my mouse disappears. All right, so what you see here, I have a model of a small protein. Okay, it's consisting of, let's say, 4 amino acids. Okay, and you see that this is a three-dimensional model, and I cannot, when I start to rotate this protein arbitrary, in arbitrary manner, you can see there is a lot of sound. Okay, so this is what Professor Ramachandran did after doing mathematical calculations. He said that because of the shape of the different amino acids, there are differences in the side chains, they cannot fold into any arbitrary shape they want, and they have to follow certain rules when they are trying to arrange themselves in a three-dimensional space. Okay, and based on his calculations, he came up with something called Ramachandran plot, which is very heavily used by the scientists who work in the area of protein structure prediction, either experimentally or computationally. So first I would like to play parts of movies which are based on his work, and then I will talk about what is a Ramachandran plot. Okay, turn off my video.
The human body, its form and shape is dependent on collagen, an essential protein found abundantly in all mammalian connective tissues such as skin, cartilage, muscle, bone, tendon, teeth and eye. Collagen is vital for the structural integrity of the body. Any imperfections or instability in its structure causes diseases such as hypermobility syndrome, osteogenesis imperfecta, and Ehlers-Danlos syndrome. 50 years ago, we did not know what caused these conditions. This understanding became possible only after the structure of collagen was decoded. It was in a small and relatively unknown laboratory in Chennai that a young physicist of C.V. Raman's heritage, barely 32 years old, unraveled the basic structure of the protein, a problem that was eluding the greatest structural biologists of the time. The discovery was to put India on the world map of modern structural biology. The young researcher was G.N. Ramachandran, or GNR as he was affectionately called, one of the greatest scientists that India has produced. When Ramachandran entered the field in 1952, those were momentous times for molecular biology. A year before, Linus Pauling had discovered the alpha helix, one of the basic structures of all proteins. The double helix of DNA, which carries the genetic code and forms the basis of all life, was discovered by James Watson and Francis Crick in 1953. But the structure of the most important protein, collagen, which accounts for 1/3 of all proteins in the body, was not known. It had engaged many top researchers in Britain and the US, Pauling and Crick among them, but the problem had proved to be complex. It was in this charged atmosphere that GNR, unknown in the field and working practically alone but armed with extraordinary intellect and intuition, had begun his experiments to determine the structure of collagen.
All right, so let's go to the next part of this movie.
1963, he, along with his students V. Shashishekaran and C. Ramakrishnan, set out to establish general criteria to determine whether particular protein conformations were acceptable or not. This resulted in what is today called the Ramachandran phi-psi plot, or simply Ramachandran diagram. A protein molecule can be characterized by two rotation angles, phi and psi, that specify the relative orientations between any two planar peptide units in the long chain. GNR's group asked the question whether all values of phi and psi result in acceptable and meaningful protein structures.
If I want to talk of protein, I must take a very basic thing. If I know how to add 1-digit numbers and carry over, I can add even 10-digit numbers. Like that, a small part of the protein has to be picked up, which can be examined. First idea, namely that the protein can now be examined in terms of peptides. Number two, the distances can be calculated. Number three, a criteria can be applied.
The Ramachandran diagram provides a simple two-dimensional graphic representation of all possible protein structures in terms of the rotation angles. The analysis was simple and elegant, but because of its apparent simplicity, the power of the technique was not fully realized immediately.
Everybody thought this is only a theoretical one, it will lie there like this. But when the first structure of myoglobin was solved and it was plotted on the map, the points just fell into the map.
But as more and more biomolecular structures are being determined, the plot serves as a confirmatory tool to determine whether a proposed structure is acceptable or not.
And that is what the Ramachandran map is now used with the globular protein structures, whereas its original thing was to use it with a fibrous protein, which number is less.
40 years after it was formulated, the Ramachandran map still remains a fundamental tool for protein conformation.
All right. So this was briefly about the contribution of Mr. G.N. Ramachandran.
63.
Just 1 second. So, if you have a protein structure, then there are two angles. One of them is called angle psi, which exists between C and C alpha atoms in a protein chain. And then the other one is called phi angle, which is between the nitrogen atom and C alpha atom, carbon backbone atom. And if you make a plot of these two, so the allowed values for phi and psi are from -180 to 180. And if you do it for any given protein, and if you find that all the amino acids which are shown here with the blue color are falling either in the core regions, which are shown here in red, or allowed regions which are shown here in brown, or generously allowed regions which are shown here in yellow, then you can be confident that you've got the correct structure of the protein. Okay. And if they are mostly falling in the forbidden region, most of the protein, that means you have not got the correct structure of the protein. So for example, many people use experimental techniques to determine the structure of the protein. Computational biologists like me also do computer simulations to determine the structure of the protein. But how do we know whether we have got the correct structure of the protein? So Ramachandran plot is one of the confirmatory tests which we can use to see whether we have got the correct structure of the protein or not. So this was the contribution of the Professor Ramachandran to the field of protein science. All right. So now what we'll do, we'll quickly change the gears and we'll talk about another problem which is called gene expressions. Okay. So if you look at the different types of cells in our body which exist in different organs, okay, all of them came from one single cell, and all of them have the same DNA. So it is the same DNA, but it is being read or being expressed differently in different types of cells, right? So we know that it is the same genetic code, but it works differently in different cells. And we roughly know that each cell uses slightly different part of the DNA. For example, cells in your eye express a set of genes which are different from the cells in your skin, okay, which are responsible for making the proteins which are required for a given particular organ. Okay. And cells can regulate the packaging and reading of the DNA depending on many factors, including external environment. And there are many proteins which are involved in regulating this. And these proteins go and bind to the DNA to regulate the gene expression. Okay. And what we can do, we can again use the free energy minimization to understand this protein DNA binding and its dynamics. Okay. So what we are going to do here, we are going to use a very toy model of protein DNA binding to understand if we have a DNA, how many proteins can bind to it. So let's say if you have a large number of sites available on the DNA for binding of the protein, how many proteins can go and bind to it. Okay. So for this, we are going to take a very simple model. Okay. And model is something like this. That suppose we have a DNA where there are, let's say, total capital N number of binding sites which are going to be available for the binding of the M identical number of proteins. Okay. And we know that DNA is negatively charged, and therefore the proteins which have to go and bind have to be positively charged. And whenever this interaction happens, it is going to lower the energy of the system, right? So let me actually go back to my iPad. All right. So suppose I have a DNA on which we have total capital N number of sites which are available for the binding of the proteins. And let's say there are total M number of proteins which are available for binding. Okay, let's say that the value of M is less than or equal to N. Okay, and we know that if the DNA is negatively charged, then if this binding has to happen, then these proteins have to be positively charged. And if they come into the contact, let's say for each of the binding, whenever this goes and binds here, the energy of the system, let's say, decreases by epsilon kb. Okay. And I would like to know, if this is the scenario, if this interaction is favorable, we have total n number of binding sites which are available and total m number of identical proteins which are available for binding. And if binding of each of the protein is going to lower down the energy by minus epsilon kBt, ultimately how many proteins will go and bind to the DNA? Anyone? Any idea? So if I apply a simple energy minimization to this problem, right? What is the answer you are going to get? How many proteins will go and bind to the DNA?
All the proteins.
So, if you apply a simple energy minimization or classical mechanics, then we get the answer that, all the proteins will go and bind to the DNA. Okay, because that when all the proteins are bound to the DNA, that will be the minimum energy state of the system. Okay, but this will not be true when the thermal energy is present. Okay, so although this is favorable, what thermal energy will do is anything which is bound here, it will also try to kick it out, right? And the how many of these identical proteins will go and bind to the DNA will be determined by the competition between this thermal energy and then whatever this energy of binding is. Okay, all right. So all of you agree with me that if I have m number of proteins and total n number of binding sites, then I can define a occupancy variable rho by m upon n, right? And the range of the values of for the rho can be from 0 to 1, right? So rho is equal to 0 when 0 sites are occupied, and 1 will be the case when all the sites are occupied. Okay, and let's say I have a particular density rho which corresponds to m number of identical protein. Okay, I can think of the rho as a macroscopic state. Why? So I can think of the state where the density is rho as a macroscopic state. Why? What is the basis for that? So a state with density rho can consist of a large number of possible microscopic states. Yes or no?
Yes, sir.
What is the reason for that? That is the correct answer, but what is the reason for that?
So because we can arrange different proteins in different.
Very good. So we can arrange m different number of proteins on these n lattice sites in different ways. Okay, and energies for all of these configurations will be identical, right? Because the same number of protein will be bound, and therefore the energies will be identical, but the arrangements will be different. So I can think of the state rho where m proteins are available for binding or bound to it. Then in that case, this is like a macroscopic state, right? And what we are interested to find out that out of these m number of proteins, how many of them will go and bind to it. Okay, so what we are actually trying to find out is that what will be the value of rho for which the free energy will be minimum, right? So that means we calculate the value of rho which minimizes the free energy. Then I have my answer that the rho which will minimize the free energy, that particular rho will be preferred. Okay, and accordingly we can calculate the density, how many proteins will go and bind in that particular state which minimizes the free energy. So if I want to answer the question how many protein will go and bind to the DNA, I have to first calculate the free energy of this entire system as a function of rho, and then I have to do the energy minimization to see which rho will be preferred, right? So let's try to do that. How do I do that? we know that the expression for the G is nothing but it is U minus TS. Okay. What is going to be the form of the U? So we can write down U, right? If M proteins are bound, then the energy will be equal to minus M epsilon kBT, right? And using this relation, we can write down that this will be equal to minus N rho epsilon kBT, right? And if you look at the dependence of U on the rho, what you see that as the rho will increase, the energy will monotonically decrease, right? Now what we have to also calculate, what is the entropy? And in order to calculate the entropy, we have to find out in how many ways I can arrange M number of proteins on total N number of sites. Okay. So in order to do that, let's try to first do a simple calculation. Okay. Suppose I have 3 sites, N is equal to 3, and I have 2 proteins which are non-identical. How many total different possible arrangements are there in this case? W, please raise your hand. If I have 2 non-identical proteins and 3 binding sites, how many different possible arrangements are there?
Six.
There will be total 6 different possible arrangements, right? One of them is this, the other one is this, second one, then third one will be something like this, then fourth one will be something like this, where 2 and 1 have to be interchanged. Similarly here, fifth one, 2 comes here, 1 comes here, and sixth one will be this, right? So in this case, we can write down in general the total number of possible microscopic states will be n factorial divided by n minus m factorial. However, if you take the problem as if that, if 1 and 2 are identical, that means you cannot distinguish between 1 and 2, right? Then how many configurations we have?
3.
In that case, we have 3, right? And in general, for m number of proteins, if you apply this, what you are going to get, that there will be a m factorial here. You can check it. Okay. So we know how to calculate the total number of possible configurations for identical proteins. Okay. Now, if you are given this, I would ask you to work out another problem. Suppose I have total 4 sites, n is equal to 4, and I am giving you 3 cases. In all of these, n is 4. In the first case, I have m is equal to 1. In the second one, I have the m is equal to 1 means there is just 1 particle. In this case, let's say there are 2 particles. And the third case, I have m is equal to 3. What is the value of these 3 cases? Please calculate and tell me. Anyone? Please raise your hand, those who can do it quickly. Okay, Faizan is again back. No, I will not ask Faizan this time. I will ask someone else. Any other volunteer? All of you have done this. So please tell me. Anyone else? Okay, Aditya. This time I will ask Madhav. Madhav, yeah, go ahead.
For n equals 1 and 2, it will be equal to 4.
Sorry, for the first one, how many?
So 4.
Okay. For the second one?
So 6.
Okay. For the third one?
So 4 again.
Okay. Let me see who else has done. Anyone else has done it? Can someone? Jay, this is fine, right?
Yes, we are considering our particles identical.
Are identical, yes.
Okay. Yeah.
All right. So, although it was a simple calculation, the reason I asked you to do this was following. What you see that when, there are total 4 number of sides and 2 number of particles, right? Here in the middle case, right? In this case, what you see that the system is half filled. In this case, system is less than half filled. And in the third case, the system is more than half filled. What you see from here, from this simple example, when you have a system which is less than half filled or more than half filled, the number of possible configurations are smaller. And when system is exactly half filled, that means when rho is equal to half, you have maximum number of possible configurations, and therefore you will have maximum entropy in that case. And therefore, if you have to plot the behavior of S as a function of rho, it will have this kind of dependence. It will be that rho is equal to half. Right? All right. Now going back to what we were doing earlier. So what we are going to calculate it for this problem, where there are total n number of sides and there are total m number of proteins. We are trying to calculate the free energy, right? And free energy we are going to calculate using this. And then for this u we have already derived the relation, and then for entropy we have to calculate it from the knowledge of W, right? Kv, L and W, and we now know the formula for W. Epsilon kBt minus T kb. This is nothing but this is n factorial divided by m factorial and n minus m factorial, right? Now whenever we have log of a factorial, which approximation we use? Anyone? If we have log of a factorial, which approximation we use?
X minus x square by 2.
No, there is a very particular approximation which is used. Probably you have studied that thing in physics. No? I'm sure some of you might have heard of Stirling's approximation, right? Yes or no? So, whenever you have a log of factorial, then this can be approximated as x ln x minus x. Okay, this is called Stirling's approximation. You apply Stirling's approximations to this problem, all right? And then what you can do, you can rewrite this entire thing. If you apply the Stirling's approximation, the final expression for the G which you are going to get in terms of rho, because n is here, m is here, and we know that rho is nothing but it is m upon n. What you are going to get is this expression for G. Okay. KB. G minus G is equal to minus n rho kBt minus kBt n rho ln rho plus 1 minus rho ln 1 minus rho. Okay, and we can write down everything like this. kBt is equal to minus rho epsilon. There was also epsilon. Rho epsilon and minus rho ln rho plus 1 minus rho and 1 minus rho. You can check it out. Okay. So this is the expression for the free energy we have got. Okay. Now, if you plot this, I have already told you that if you look at the dependence of the U is going to decrease like this, And the other thing which is there, s as a function of rho is going to behave something like this, right? And if you plot the g as a function of rho, this is how it would look like. I'll show you the plot. Okay, so if you look at the plot of the g as a function of rho, okay, let's say this one is for epsilon is equal to 2, then you see that s is behaving like this, okay, as I've told you. And the reason is that when the system is exactly half filled, the entropy will be maximum. You will decrease monotonically, and for the value of epsilon as 2, the g will be minimum at the value of 0.88. The value of rho is 0.88. Okay. However, if the value of epsilon is 0, that means there is no energy cost involved in the binding of the protein to the DNA. It can happen spontaneously. Okay. If this process is spontaneous, in this case, the value of rho that will minimize the free energy, that value will be, this becomes e, if epsilon is 0, this becomes e to the power 0, 1 plus e to the power 0, and then rho is equal to half. So again, you get back the previous result, that when the reaction is spontaneous, when there is no energy cost involved, then in that case, system would like to go to that state where the entropy is maximum. And here I know that when rho is equal to half, only in that case, the entropy of the system is maximum. Okay. So again, even in this problem of simple protein DNA binding, we see that we are again getting back the same result which we saw earlier, that when there is when the reaction is spontaneous, when the process is spontaneous, if we consider the process of protein DNA binding to be spontaneous, then in that case, system would like to go to the state where it can maximize entropy, and that in this case will be rho is equal to half. Okay. And if there is an energy cost involved, then the value of rho will be something different. Okay. For example, it is shown here for epsilon is equal to 2. So summary of what I told you so far, that proteins that bind onto the DNA control the gene expression in cell, and then this protein DNA system minimizes free energy, and number of proteins bound to the DNA will depend on the free energy of the protein DNA system. Okay, that is what we found from this problem. All right. So this is the summary of today's lecture, that we discussed the model of protein folding, we discussed the Ramachandran plot, and finally we looked at a simple toy model for protein DNA binding. Now, I would like to give you some of the take-home messages from this particular module of the BB101 course. So what we saw here in this module in the beginning, that if we are talking about the microscopic world, like the world in which bacteria live, the world in which biomolecules live, the inertia is negligible, and that's why the protein takes a lot of time to sediment. Okay, and these bacterium don't have any inertia. However, one of the important things which I told you is that you can still use a combination of mass, spring, and dashpot to understand some biological systems and phenomena, and we applied it to two problems: bacterial swimming and sedimentation of the proteins. After that, we saw that the swimming of a bacteria is different from the swimming of a fish. Okay, so for the bacteria, the motion is at the low Reynolds number, where viscous forces dominate the motion. Okay, and one of the things which I told you, that we can take the advantage of our inertia, or a big fish can take the advantage of the inertia, but the bacteria cannot take the advantage of its inertia, and therefore it stops as soon as it stops moving its flagellum. Okay, so motion at the low Reynolds number world is very different from the motion that we see in the high Reynolds number world. And a consequence of this was the Scallop theorem, which tells you that at low Reynolds number, microorganisms cannot swim by executing a geometrical reciprocal motion, and that's why many microorganisms like bacteria, okay, have flagella, okay, and some of the microorganisms also have cilia, and if you look at their motion, they do not perform a geometrically reciprocal motion, which refers to any motion where the moving parts take the same path while going in the forward and backward stroke. Okay, and since we know that due to geometrical reciprocal motion we cannot make anything to swim in water, and geometrical reciprocal motion is the one which is easiest to implement if you are fabricating something at the microscopic scale. And if you have to make something to swim by executing geometrical reciprocal motion, in that case you have to replace the fluid by a non-Newtonian fluid. After that, we looked at the thermal forces and Brownian motion, and what we found that they are thermal forces which are responsible for the Brownian motion. And one of the important things that we learned in this module is that if you look at the thermal energy, then the magnitude of thermal energy is very, very tiny, and therefore in the macroscopic world we can ignore the thermal energy. However, the moment we go to the scale of the biomolecules, what happens that this thermal energy becomes comparable to the other forms of the deterministic energy. Okay, and since the different forms of the deterministic energies are also identical at this length scale of biological molecules can very comfortably convert one type of energy into the other type of energy. And the other things that biomolecule can do, they can simply utilize the thermal energy and they can go under a shape change, which doesn't happen in the macroscopic world. Okay, after that we discussed diffusion, what is diffusion, and we derive the diffusion equation which connects the change of concentration with space to the change of concentration with time. One of the very important thing which I told you about the diffusion is that in case of a ballistic motion, generally the displacement is proportional to t. However, if something is going from one place to the other place by the process of back and forth movement, then the x grows as to the power of half, and a consequence of that is that diffusion becomes a very, very slow mechanism over a longer time scales, and that's why our cell cannot utilize the mechanism of diffusion for transporting things from one place to the other place inside the cells, and they use active transport which is done by the motor proteins. After that, I talked about the contribution of Einstein to the field of Brownian motion, and I told you about the Einstein relation which was derived by Einstein, which was gamma d is equal to kVt, which tells you that at thermal equilibrium the product of drag constant and diffusion constant is a constant quantity. If one of them decreases, the other one has to increase. However, one of the important thing which I told you was that this theory of Brownian motion by the Einstein gave a very strong support to the notion that atoms and molecule exist, because in year 1905 it was still debatable that whether whether atoms and molecules exist or not. Okay, and using the Einstein relation in year 1905, one could calculate the Avogadro's number. After that, I told you about the reaction diffusion system. So diffusion in itself is very interesting, but when it gets combined with the reaction, it becomes a much more richer system. And Alan Turing, whom we know as a very good computer scientist, very good mathematician, he was also a pioneer in the field of reaction diffusion system, and this was something which was used by the scientists in past to understand pattern formation in nature. Okay, after that we saw that if the thermal energy is present, which is generally the case at the microscopic scale, biological systems try to minimize their free energy. We learned about the concept of macroscopic state and microscopic states, and then we learned how to calculate entropy from the knowledge of the microscopic states. Then we looked at the toy model of protein folding, and we looked at the importance of Ramachandran plot, and today we also looked at a toy model for protein DNA binding. So this is the end of the physical biology module. I hope that you have found the physical biology or biophysics exciting. You realize that you can use the physics and mathematics you learn to think about biological problems. So my request to you is that every time you see a biological phenomena, think how you can use your science and engineering knowledge to understand it, because we know very little about what is going on in many biological processes. So there is a great opportunity for all of you to go and make important discoveries in the field of bioscience and bioengineering. With this, I would like to thank you for attention that you have given me, and I will stop the lecture recording, and I will take any questions that you may have. Right? Just give me a second.